All right, guys, let's go ahead and get started here. Crazy week, I know. We missed half our labs. Just to reiterate, for anybody who wasn't aware, didn't read the emails and such, we won't be doing the GRIMS, the advanced staining, the special stainings, which is the endospore staining and the acid-fast staining. We did also do the negative stain, but you don't need to do that. Just keep the data around for that. Don't do anything else with it. You don't have to turn it in, anything like that. For next week, we're going to move on with what's scheduled for next week, just because we're trying to keep things relatively in place and more or less stabilized, so just keep that in mind. If we make any changes other than that, we'll let you guys know. So with that, let's talk about what's coming up next week, which is transformation and isolation. I don't know why it's not even that dark. Okay, anyways. Transformation and isolation of bacteria, among other things. A couple of important key things. Make sure you disinfect your work area, as always. Make sure you guys don't carry any tubes in your hands. Use rack at all times. If anything breaks, like a slide, which isn't going to be very relevant here, but just make sure you let your TA know. Notify your TA of any spills. Do not handle back incinerators directly. Make sure you guys turn them off. Gloves, goggles, lab coat should be worn at all times while you're in lab. And we will be using a BSL-2 agent technically, so just keep that in mind. So we're going to talk about transformation first. So what is transformation? It's the process of horizontal gene transfer with bacteria by picking up genetic material from the environment, which is a really vague way of explaining it. But basically all it means is we're taking some DNA from some other source and putting it into the bacteria to change it. Now, in this specific case, there's a couple of ways you can describe that process that I just did. One of them is through transduction, which is through phage, which we'll talk about in a second. But generally speaking, through transformation is where we have DNA from the outside environment and somehow shuttle it into the cell in some method. One of the famous experiments was with, trying to figure out what causes, what transfers, what's the genetic material, the material that transfers genes. Is it DNA, is it RNA, is it protein? And we were able to figure that out, of course, through this wonderful experiment where we took rough, non-virulent bacteria, in this case, I believe it was Streptococcus pneumoniae, inject it into a mice, mice is fine. Take the smooth virulent type, inject it into a mouse, mouse dead. Okay, makes sense. Heat kill the smooth virulent type, mouth's alive. All right, makes sense still. Mix the R type and the S type, which is the rough, non-virulent type, with heat-killed virulent type, mouse die. That's weird. And so what we were able to determine from this is that, and in part, this experiment's actually really successful, in part by dumb luck, which is this bacteria does like to take up DNA from its environment rather readily. Not all bacteria do that. Some bacteria are exceptionally picky and will not take up DNA from the environment. In this case, this one does. It's greedy. It likes to take up stuff that it randomly finds and either incorporate it into its own genome or make a little plasmid, and that's what happens in this case. So it was able to take up DNA from these dead guys, stick it in itself and say, hey, I got a new gene, and I'm going to use it to kill mice. So there you go. So that's a general concept of transformation. That was the classic experiment to show that. It was actually a really interesting experiment just because it taught us so much, and yet at the same time it was only discovered kind of by luck because the bacteria that we chose was correct. Fluorescent proteins. So one of the things that we're going to be messing with is fluorescent proteins for this experiment. It's basically scientists' arts and crafts show. It's not really. We use the different fluorescences for various properties, especially if we're trying to verify different types of locations of proteins. And if we want to locate one or more proteins, we use different types of fluorescent colors, wavelengths, that let us visualize it easier. And the really tricky part, the actually really, really interesting part, is a lot of these are basically all very, 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 very similar proteins with small modifications to them that create a slight variation of color. So the FP1 and the GFP, these are all basically the same protein with just small little changes to them, little tiny ones, not even that big. But they do give us a wide color range. You can see that the RFP ones generally give us this nice red color. And the green ones generally give us a nice green color, but it can go pretty far in different directions. This is one that somebody created to kind of show off a bunch of different types of bacteria. Made a nice little luminescent painting, which is great. We will not be playing with anything this fancy right now, but maybe later in the semester we will. We'll see. So let's talk about different types of transformations. So there's different ways you get genes into bacteria. One of them is with a gene gun, which does not look cool like this one, but instead just looks like a little needle that blasts stuff into a cell. It is by far incredibly boring looking, but it is actually kind of a neat methodology. Basically, we take a cell and we go, hey, let's put some DNA into you, and we can attach the DNA to gold nanoparticles, put them under a high burst of air, and then basically have them puncture the cells, and the cells that survive and are able to close back up the holes now have this DNA inside of them that they didn't before. And this is actually really, really handy for a variety of different cell types. We used to use this a lot more than we do currently because the current methods are a lot better and we have better methods overall to make these kind of modifications. But at the time, this was like one of the only ways you could do this. It's also really helpful for cell types that are extremely resistant to kind of modification. So they have like plant cell types and things like that, things that have a lot of rigidity to them that make it difficult to alter the tissues. So it's actually really cool. Very, very cool. Not done as much anymore, but still really neat. The other one, electroporation, where we basically take a cell, we stick it in a solution, we put some DNA in that solution, and then we put an electric field through there. And the electric field, because the DNA is charged, will slowly but surely kind of want to move through, and it'll just kind of work its way past through the cell membrane and into the cell. And if you kind of like stop it at the right time, the DNA is inside the cell. Worked pretty good. It wasn't amazing. It didn't work for all cell types because a lot of them are really finicky about, being shocked for long periods of times. And also it doesn't work for a lot of cell types that have very thick protein matrices and that kind of get in the way of this. But it does work to an extent. It's really handy. Also, the other downside to this is that it's not the smoothest of all things. So you can actually get leaks a lot. And leaks don't sound like a big problem, except that the voltage for this is super high. So if you touch the leak while the thing's running, you can die, which is bad. So generally speaking, it has a couple of downsides to it. The last one is bacterial transformation. And there's a number of different ways you can do this, but for the most part, it gets done through chemical means. There's some other ways you can kind of do it, but basically just chemical transformation. So the chemical transformation, basically we destabilize the cell wall of the bacteria. We say, hey, bacteria, let's just kind of loosen everything up, make you a little bit porous, maybe not be as stable as a cell anymore, and then we just introduce some random bacterial DNA, or maybe mammalian DNA, or maybe viral DNA. Doesn't matter. I mean, it does matter to an extent, but basically we just allow you to be more freely accepting of this outside DNA, and then we kind of chill everything back down, help you re-solidify, and hopefully you've got some genes trapped inside you. And then we just select for those, and we'll talk about selection in just a second. But basically that's the three major types. So gene gun, electroporation, chemical transformation, and we're gonna be working with chemical transformation in the lab. Okay, so cool science thing of the week. This is actually a really interesting one, which is something that's not transformation. It's called transduction, which is the use of phages to transfer genes to a bacteria. Now, when phages do this, this is typically, where they basically are trying to kill bacteria, but occasionally the bacteria incorporate some gene that was on the phage into their own genome, and there you go. So that can kind of happen. This is actually a really, really interesting study, which again, happened by chance. These guys were studying something completely different that involved phages, and what they were doing is that they were fluorescently tagging, they weren't fluorescently tagging, they were making these bacteria produce different types of fluorescent chemicals. In this case, they were making these cells, these R3 cells, produce a gene that made it glow green, and they were mixing them with these other types of cells, which are S cells, which are susceptible to this type of phage that they were looking at. And because in nature, a lot of times you get a lot of cohabitation of different strains of bacteria, because there's rarely every time you get a pure culture in nature. So they were just kind of looking more at this, studying it in the real world, what would happen with phages. And so they had a resistant type and a susceptible type. Then they had a phage that was producing a little tiny bit of red fluorescent protein. And so you can see these phages latching onto these guys here. These guys, these nice red guys right here, the phages don't stick to them at all, so they don't have any of the red part, they just have the green part. And then they did a mixed culture. And so you can see the little red guys right here and the uncolored guys right here. These are the susceptible cells. They don't have any GFP on them. So you can see them all mingling together. But then they noticed this guy right here, and that doesn't look right. How is a phage attached to this guy? And so they were like, that's weird. So what they did is they're like, okay, we're going to expand our study. And the reason that they were doing the study originally is from this scene right here, which is where they were co-mixing these guys, and they were showing the phage infecting mixed ones, and the mixed ones would get the GFP, start producing GFP along with the lysis protein, and then they'd lyse themselves, so you could see those lysing. But they also noticed that some side bacteria were also lysing at the same time that were supposed to be resistant. So what do they do? Well, they studied this. They basically dropped what they were doing, they switched over to this completely, and what they were basically able to figure out is, as these cells that are susceptible were dying, they would still have some phages attached to the outside of them. And when they kind of died, they'd bleb off a little chunk of themselves called a micelle, which I'm sure you guys have heard. It's just a small circularized bit of lipid, and sometimes on the outside you have receptors that viral particle can bind to, or sometimes it's attached. So here's an example of that. You can actually see it. This is a little micelle from a susceptible guy. So it's blebbed off, and here attached to him is the viral particle. Pretty cool. And what they were able to determine was that if you just kind of like let micelles kind of float freely in nature, occasionally they'll just kind of like run into an actual real cell and just kind of bleb into it and just join part of its whole cell structure. And that's basically what was happening. So they were basically able to infect these resistant cells with a phage that they were resistant to because they were cohabitating with cells that they were susceptible to, because they were getting those little blibs coming in and they'd be able to kind of mesh into them. So it was actually a really interesting study. It's actually a really funny thing. If you guys look online, although I'm sure you guys don't care that much, but they did a bunch of interviews and they were like, yeah, we were like totally not studying something else. And they suddenly found this thing that nobody had ever hypothesized happening. They didn't know happening. It's a completely new type of phage infection, basically by these micelles. So really kind of neat. They also included this little video, which I'll show you guys here. It's actually kind of neat. So these are cells that are currently getting infected with phages. And as these cells are increasing in luminosity, that means that they're lysing because the phage is also encoded a GFP, fluorescent protein. And so you can see anything that lights up is going to get lysed eventually. This is actually just kind of a cool little video showing how quickly this happens. I think this is over a 12-hour period. So it's not amazingly fast because it's obviously only a 10-second video, but it's still pretty fast. So take-home lesson from this. What can you take home? One, you should be able to take home that fluorescent proteins fluoresce. They're really easy to track something. Two, you should take home that cells that are resistant to a certain type of phage can actually gain susceptibility by cohabitating with susceptible phages, or susceptible bacteria because of that little micelle action. And three, always keep an eye out for small problems in an experiment because you never know. You might discover something completely new that's completely different from anything anybody's ever seen before, which is what happened here. So really cool little study. Okay, enough rambling on that. So we're going to talk about transformation. This is our general transformation flow of what we're going to be doing in lab. And I think there's a problem on Lab Archives because they've got some numbers changed on Lab Archives. This picture is the one that you guys want to follow. I'm going to follow up with Dr. Lawson to see if we can fix that. But basically what you guys are going to do is you guys are going to get a sample of E. coli. This sample of E. coli you actually are going to be making on Monday through the Cultural Transfer Techniques lab, which we'll talk about in a second. And you guys will take a colony from here. You guys will swirl it around in this mix of calcium chloride. So calcium chloride is going to be the chemical thing that helps shuttle the DNA in. So it's a nice and handy methodology of getting this in. So we're going to add calcium chloride to a tube that's labeled positive and negative. We're going to put E. coli in both positive and negative tubes. Positive and negative refers to the next step after this. From these tubes, we're going to plate each of them on a plate that's either that's just LB, which stands for Loribroth. So Loribroth plate, or Loribroth plate with ampicillin. Ampicillin is a selective antibiotic that we use oftentimes to say, okay, do you have resistance to ampicillin? We expect bacteria to grow in this plate right here, but we don't expect any of them to grow right here. We want to double check that these bacteria are all susceptible to ampicillin, because that's our selective marker for our plasmid, which is P-glow. We'll then have a tube that's got P-glow and a tube that's got water. We will actually aliquot out for you guys the amount of P-glow you need, so you'll have a little tiny thing. If you guys tip it over, you'll have dumped out all of your samples, so please don't do that. So be very careful, because it's only 10 microliters. You guys will put all of the P-glow from here into the positive. You'll take the water, that'll be our control, into the negative, and you're just going to let it chill under ice, so in ice conditions, for about 15 minutes. After 15 minutes, you guys are then going to take your tubes. You are then going to go over to the water bath. The water bath is going to be set to 42 degrees Celsius, and you're going to dunk them in for about 50 seconds. Double check when you guys put it in the water that it's set to 42, that the temperature reads 42, because if it doesn't, you're going to have problems. It can't be higher because it'll kill the cells, and it can't be lower because then it won't activate it won't basically make the conditions good for allowing the DNA to go through the cell walls. After 50 seconds, you're going to cool it back down on ice for 1 to 2 minutes. So you're going to let it sit down here in ice for 1 to 2 minutes. And the reason why we have a recovery phase is because heat shock is actually stressful for the bacteria. 42 degrees Celsius doesn't seem like a lot, but if you guys had a temperature of about 42 degrees Celsius internally, you would feel really, really, really unhappy. So just keep that in mind. Bacteria, same way. So it stresses them out. You need to give them a chance to recover. Then we're going to add some warm 37 degrees Celsius broth. We're going to let them chill in the incubator for 10 minutes in that 37 degrees Celsius broth. That gives them good conditions to grow again. That's basically saying, okay, you guys chilled out, you've re-solidified your cell walls, you guys should be good now. Here's some nutrients, here's some good temperature. Maybe start recovering and being set up to go again. Then after the 10 minutes, you guys are going to streak them out again on a Loribroth plate and another Loribroth plate with ampicillin. Now, the reason why we do it with this one is because we want to just check to make sure that they didn't all die from this step. They probably wouldn't die from this step, but they definitely might die from this step. So do they still exist? Then we're going to do a Loribroth plate with the ampicillin. Only one of these guys should grow on this plate. Now when you guys do the streaking for these plates, it's like this. It's kind of a weird one. So you streak down the center, and then you streak up and down like this to kind of spread it out because it's on half a plate, so be careful with that. Don't cross over accidentally. You're going to get weird results. So what do we expect to have happen? Well, we've got ampicillin, which should kill our bacteria normally. Makes sense, pretty easy. Ampicillin kills things. We're going to have our plasmid. We're going to put it in there through the transformation. This plasmid has a gene on it for ampicillin resistance. So only bacteria that have the plasmid should be alive on there. So that's why we're using that as our selective agent. On this plasmid, we don't just have ampicillin, which is the ampicillin resistance gene, which is basically shutting down the effectiveness of ampicillin. So it's binding it and basically degrading it, so it's not working as well on these cells. But we also have Ara-C, which is an arabinose binding protein. And it is going to come in here and bind this thing called PBAD, which is the operon, the promoter that says, hey, come in and make this gene, which is our fluorescent protein, GFP. So it's sitting here and it's going, hey, I'm just going to stick on here and not do anything, and I won't let you make any copies of GFP unless you have arabinose present. So don't worry about the arabinose part as much, but basically just know that this is under the control of arabinose. So this will only happen if there's arabinose on the plate, which we're going to take care of for you guys, so you guys don't have to worry about. But once that is there, then it'll be able to produce this nice little fluorescent protein, which will fold and make a nice little glow for us when we put it under UV light. So the only ones that we should see grow after we do the whole transformation plate on an ampicillin plate is the one that has this plasmid, and those ones should glow. That's the only ones that we expect to do there. So it should look something like this. So you can see that there's a bunch of bacteria growing on, oops, why did it jump back? This plate is a Loria broth plate with no ampicillin and no arabinose. So you can see that you've got bacterial growth on both sides, but there's no glowing. That's because there's no arabinose on here to make the glowing happen. On this one, this is a plate with ampicillin and arabinose, so you can see that there's only a few colonies that are able to survive on here. You actually get very low copy number on this because transformation isn't a really super clean thing, so you're only gonna get a few colonies on here. This plate has ampicillin and arabinose, so you can see these guys, this side is the one that's positive for the plasmid, so you get the glowing colonies. These guys right here that you can see on this side theoretically should have been the control side. There should be no plasmids on there. This is the trick though, because we're adding liquids, if you're not very careful, liquids will spin around the edge, and that's what happened to these guys right here. This is the reason why. So you can see how they're really close to the edge and not in the middle like these guys. That's because there was a little liquid right here and it just went whoop, and it sped around the entire thing and you get a little contamination. So contamination, kind of a big problem here. So take your time, be careful. What we will do on day one is we will do the isolation, which we'll talk about in a second. On day two, we'll do the transformation, but on day one, we'll also have a little puzzle here to do this image. So you guys kind of get an idea of how this is supposed to go, so you guys get that locked in your brains really well, and then we'll do the rest. I shouldn't have made so many clicks for that. Okay, does anybody have any questions on the transformation end? Makes sense. It's complicated, I know. If you guys do run into questions and you're like, I don't understand how this works, make sure you guys let us know. We'll talk it over with you guys. Now the next part, which is isolation of pure cultures. This is a technique-heavy thing. It's very similar to the other streak for isolation, but in this case, we're looking for an isolation of a pure colony from a mixture. So subculturing, just in case you don't remember, isn't just about subcultures within humans. It's, even though bacteria also do go through an emo stage, it's the process of transferring bacteria from one media to another in an active growing state. We also use it to help us isolate a single genus and species from a single colony, because if we see a single colony, we say that was probably just one bacteria. And we've talked about this before, how to do the streak plate isolation. We've done it with pure cultures before in the past. This time we're going to be doing it with mixed cultures. This is where this comes in much more crucially, to make sure that you guys are going the right directions in here and you're not accidentally crossing back over. If you have too much bacteria on here, you're going to get bad results. What we're going to be doing, just as a reminder, you guys will get a loop full of your mixture. You will streak it here on this end and just go up and down in a zigzag pattern like that. You'll flame your loop, you'll let it cool down again, and without adding more bacteria, you're just going to go across this here. So you're not adding more, you're just lowering the numbers. This becomes really, really important because some bacteria will grow at different rates. So you might only get one isolated colony of one type here and one isolated over here. So we have to be very careful when we're doing this transfer. We want to make sure we take our time. Rushing is bad, take your time better. Also, gouging auger is really bad for this. If you gouge the auger, especially in the second or third quadrant, all the bacteria will be wiped off your loop effectively if you gouge it, and they won't work very well. So make sure you guys are really careful with that. We're going to be giving you a mixture. The mixture is going to contain two types of bacteria. One of them is your E. coli, and the other one is going to be a mystery bacteria. It's not really a mystery, it's Serratia marcescens. But basically these two bacteria are very different from each other in looks if you grow them under the right conditions. Key thing here, we're going to incubate these and grow them in a room temperature incubator. That's because Serratia only produces a dye at room temperature. If you guys grow it at high temperatures, it will look kind of the same as E. coli, which is not very helpful. It's not perfectly the same, but it's very similar. When you guys actually do this, the point of this is to observe cultural characteristics. There are many cultural characteristics you can determine just from plates. Among them include the following in this chart, which are pigmentation, form, elevation, margin, and colony size. And I'm just going to go over them really quickly. Pigmentation is just like, what is the color of the bacteria? It can be anything from pure white to slightly tan to pink or red or slightly green, all sorts of variations. Purple even. Form is basically the shape of the colony itself. You can have little tiny dots, which is punctiform. You can have large circles, which are just circular, so they're perfectly smooth all the way around. You can have things that are filamentous, which kind of have these little tiny threads moving out from the center of them. Less common, but it sometimes happens for our lab. irregular, which is just kind of a not circular shape, so kind of wavy throughout the entire thing. Rhizoid, which kind of looks like a lightning strike or roots coming out from it. And then spindles, which are kind of like little tiny eye-shaped ones. Don't worry about those ones too much. The other ones, the other parts are things like elevation. And I'm not going to go into detail about all of these, but I will talk about these guys real quick because they're kind of hard from the description. Elevation, if it grows completely flush with the auger, that is considered flat. So it'll be flush with the auger, it won't have any height to it, it'll just be basically in line with the auger. If you have a little bit of height, so it kind of looks like a mesa or a plateau, it'll have a little bit of height, but be flat on top. So it'll be rising above the auger surface just a little bit, but it'll be like almost as if you like stuck a piece of, I don't know, ham on top of a dinner plate. You can see it rise above there, but they're both flat on top. Okay. Convex is, much like the name, for an eye shape, it's got a hump on the top. So it's just a perfectly nice little bump right up on the top, so it's convex shape. Pulvinate, very similar, but it's a very exaggerated hill, almost like. So whereas this is just kind of like a little drop of water, this is kind of like a big bead of water, like maybe a hill that you were trying to walk across. So this one's just kind of like a gentle slope, this one's a big hill. The last one is umbonate. Umbonate's kind of obnoxious. Basically, it's easily described as something that's raised, so it has an elevated surface that's flat, but in the center of it is kind of like a little colony sticking out that looks like these convex or pulvinate ones. So it's got just a little bit that's sticking out there. So if you imagine that you've got a piece of ham with like, I don't know, an olive sitting on top, that's what it's like. Just like a little bit of a bump there. You will see umbonate in lab from some bacteria that we have. It does happen quite a bit, but the descriptions in the book are sometimes difficult to see. And the other ones are pretty much easy to kind of go over. So you guys will be, oh yeah, I forgot I put a zoom on that. These other ones you guys will see, mostly the things that are at the top are the really common ones. Like this, not common, probably won't see in lab. These, very common, very large, not as common, you won't see in lab. But you need to get good at looking through these. These are good cues and indicators that these are the growth characteristics of a certain bacteria that you're looking for, so it helps you differentiate them. Word of warning though, cultural characteristics are not universal for all bacteria at all times, and a great example of this is one of them we'll be using is Serratia marcescens. These are all examples of cultures of Serratia marcescens. You can see that these guys right here are very bright red. These guys right here are very bright red. This right here and this right here are not red at all because they were grown at a higher temperature and they do not produce the pigment at a higher temperature. So you cannot rely on cultural characteristics for everything or even like the pigment for everything. So they all grow different depending on conditions like time, temperature, humidity, type of media you're growing them on. But serratia is actually pretty easy. If you guys look at it right here, these little edges, these are little teeth. That means it's serrate. Thus the name serratia. So they're serrated teeth on the edges. It's got a nice glossy pigment that's kind of moist. It's motile, so it'll move through media rather nicely, so making it cloudy. Same thing for the broth over here. The other one we'll be doing is E. coli, which we've seen before. This E. coli is special, of course, because it is E. coli that is susceptible to ampicillin. And this is the E. coli that we're going to be using for our transformation. You can see it's got a nice tan coloration. It is not serrate. It's very smooth on the ends. It also is motile, so it makes everything cloudy in a sim or a broth. And you can see it's kind of got an undulate shape here. So not a very smooth shape, but it doesn't have the teeth like serratia does. What you guys will do, you guys will have stock cultures. When you get cleanup for that, you'll just make sure to put the stock cultures back where your TA tells them to. They might ask you to save them. They might ask you guys to put them on the autoclave cart or in the biohazard bag. If it's biohazard bag, make sure the plates go in the specific location. If it is the autoclave cart, if it's a slant or a broth, make sure you guys put it in the right rack. They will remind you guys which one it is. For your plates that you guys inoculate, make sure you guys put the plates in the specific incubator slot for your lab group. So these are set to room temperature. You guys want to make sure to put them in the incubators that are room temperature incubators. On results day, when you guys get done with the plates and you've done everything you needed to for Wednesday, you will dispose of the plates in the biohazard bag. Make sure you guys use your plates for your transformation process first, but after you guys are done with them, you can dispose of them there. As always, you guys will sterilize your cell phone bags with Cavicide, your countertops with Cavicide, wash your hands, and you'll have a wonderful day. Real quick before you guys leave, and we're still good on time, which is great. With the transformation lab, we are going to be transforming it on Wednesday. We will keep an eye on it over Friday and the weekend and select the best plates. And we will have those plates for you guys to view on Monday. So lab report will not be due until Monday next week. I think in Canvas it's set for a little bit incorrectly. I'm going to update that in Canvas, but just be aware that your lab report for the transformation lab will be due on Monday. The lab report for the cultural isolation lab will be due on Thursday next week, okay? Any questions on anything I covered so far? Everything makes sense? All right, I will see you guys next week. Have a great weekend. Come see me if you guys have any questions, and I will talk to you guys later.